Good morning. Good morning, everybody. Welcome back. Welcome, everybody. It's nice to see your faces again. All right. Well, welcome. Good morning. I feel you, Shane. All right. Keep logging on to Pear Deck. We're going to use that a lot today. And once again, we transition. Change is the constant, right? So we'll keep doing what we can do. I do have a brief check-in for you. Originally, it was using the mood meter to do a practice around loving kindness, but actually, I think is more appropriate before the Thanksgiving break. So I've got something shorter for us this morning that can actually have you reflect on what happened over these last week since we've been together. And then we're going to begin. Actually, I'm going to ask you a question. It has been so long since we've been together. Some review of what we were doing when we last were together would be helpful. Got some practice questions for around those concepts with ATP mediated energy transfer. And then, before break probably, but maybe you watched it during break, we're going to begin to transition into some biochemistry and thinking about if there are catabolic and anabolic processes, right? Processes that release energy, those are the catabolic ones, and processes that absorb energy, those are the anabolic reactions. Then what do those reactions actually look like in biology? And begin to look at not just what those reactions are, but the functional groups, the parts of the molecules that are specifically reacting with each other as molecules form or as molecules come apart. So it was an Edpuzzle video on functional groups that I made that I hope you had a chance to watch. Maybe you watched it before break, and that's okay. You can always go back and watch it again if you wanted to. I've got some practice work with functional groups and then with some intro biochemistry for us today. But as always, it's been a while. What questions do you have before we get started this morning? I'll keep the chat up just in case people have questions they want to put in words. But if there's no questions, we'll go ahead and get started. All right, so my check-in is really simple this morning. As you think back over these, 5 days of break, but again, we haven't really seen each other for a week. Can you choose a word that best represents your experience over these last 5 days, 6 days, 7 days, whatever you choose? You can write it, you can type it, doesn't matter. This might just be a way for us to reflect a bit and then to look at what people's experiences were over these last few days. Just take a minute or two, maybe think for a bit, and then type or write whatever word, and it can be more than one word. But a word or words that best represents your experience. Looks like just about everybody has written something. Give you a few more seconds to finish your word or your words. And of course, you don't have to celebrate Thanksgiving. We all hopefully had a break from school. All right, let's take a look. Sleep. Incomplete. Interesting. Yeah, weird. Wasn't it weird? Simple. Stressful. Interesting. Restful. Filling. It's always nice to have food. Lonely. Wow. Hectic. Fun. Frazzled. Yeah. It felt off for me too. Stressful. Relaxing. Lots of different experiences here. Another fun. Normal. I like it. All right. So, kind of runs the gambit, right? But maybe this simple experience just gives you a chance to see that your experience was not unique. Or maybe it was unique, right? It doesn't really matter. But it's always good to take a moment to reflect and think about what our experience was. For me, I agree with weird for sure. Separate, or there weren't any big family gatherings on my end. And that was definitely different. But also, I think the responsible thing to do. So that's my 2 cents, if I could type a word or write a word. All right. Well, thank you for doing that, as always. I hope just taking a few moments to be reflective is helpful. We'll go on to the biology. And as I mentioned a few minutes ago, it's been a while since we've been together. So I have a practice free response question around ATP-mediated energy transfer that we can take a look at. So if you think that would be helpful to you this morning, if you could just drag your dot to either the thumbs up or the thumbs down. I think I know how you're going to respond, but I want to give you the chance to respond regardless. If you don't have the Pear Deck code and you need it, remember it's in the upper right-hand corner, CJVBLZ. All right, real quick. Looks like most people are in the yes category for doing a practice question. Okay. If you're in the no category, that's totally fine. You can take these next few minutes to do something which benefits you. It's totally fine with me. But if you think a practice question would be helpful, this is the other practice that we're not going to do today. Let me make sure. All right. So here is the practice question. Again, it's an AP free response question. So give yourself an opportunity to read the background up top, study the figure down below, and then when you're ready, you can type your response. And I am, of course, here for clarifying questions if that would be helpful. Obviously a reaction you've never seen before. You don't know much about it. You don't have to. About 2/3 of people have added a response. Try to get your response in. It's okay if you're not sure. There's no grade here. This is practice. It's review. I have a scoring guide that we can take a look at together. It's kind of frozen at 12. Does that mean 6 people just aren't going to add a response? Just so I can get a sense if you do need some more time. If you have your camera on, you can show me a finger. Maybe not the middle one. But if you don't, in the chat, just how much more time do you think you need, minutes wise, to add your response? Yeah, fist is 0. Thank you. I got one more. Good. Folks who don't have their cameras on, just pop something in the chat. 0, 1, 2, 3. Very, very helpful. Really just to show me that you're still there, if nothing else. I hope you're still there. All right. Well, I'm not seeing a lot of non-zeros. So why don't we first take a look at the scoring guide, since this is an AP free response question, and I can kind of we can go back to the question and take a look at how the scoring guide applies, but also, of course, how our learning from last week applies to hopefully your ability to answer a question like this. All righty. So the scoring guide actually is really simple for this particular question. It simply says that energy is being released as ATP is being hydrolyzed, and hydrolysis is just a form of catabolism, so it's ATP breakdown, and then captured in the formation of the new bond between F6P, fructose 6-phosphate, and the phosphate to form fructose bisphosphate, or fructose 1,6 bisphosphate. So my hope is you're seeing the parallels to that really simple model, that albeit a virtual model that we were looking at last week. This idea that ATP in its triphosphate tail can be catabolized, that third high energy phosphate group can be removed, but that when that phosphate group is removed, there's some magnitude of energy that's associated with that phosphate group. Of course, ADP, what's left over, has some energy associated with it. But that phosphate group, upon being removed, has energy associated with it, and then can be transferred to, I think in our model, we just called the molecule X. And that when that phosphate group was attached to molecule X became energized. It changed its shape as a consequence. That was a way to begin to think about how a molecule might do work within a living system. It's accomplished through energy absorption and then shape change. But really what's most important about this is that that phosphate group is carrying energy with it, transfers it to molecule X becomes energized and is thus a more highly energized molecule as a consequence. So if we were to go back to the question, we're seeing a lot of that in this reaction. Again, it's a reaction that you've never seen before. And becoming more comfortable with the reality that the AP exam, heck, our exams for that matter, are going to present you with scenarios that you've never seen before is really important. You can't memorize your way into or out of AP biology. It just doesn't work. You have to gain comfort and ease with the unfamiliar. So here's this molecule of fructose 6-phosphate. Okay, that's our reactant. And this is obviously an anabolic endergonic reaction. We can see the ATP is being catabolized, right? What isn't clear necessarily, but is happening as you look at the diagram, is that the third phosphate group from ATP is being transferred from ATP to fructose 6-phosphate to make fructose 1,6 bisphosphate. And that phosphate group is sitting up there. So the energy has been transferred from one molecule, ATP, to another molecule, fructose 6-phosphate, to make a more highly energized molecule, fructose 1,6 bisphosphate, due to the transfer of that phosphate group. Okay, so you could even come in here, right, and draw an energy diagram where you could have something like ATP as a reactant, and in an exergonic reaction, we've got the catabolism of ATP. But we have another reaction, can even change colors here, that's taking place. Something like this, where fructose 6-phosphate is being formed to make fructose 1,6 bisphosphate. Where's that phosphate group coming from? It's coming from ATP. But upon it being transferred from ATP to fructose 6-phosphate, the resulting product has more chemical potential energy associated with it as a consequence of that phosphate group being transferred. And if you look at the scoring guide, right, the College Board is saying, well, the energy is stored in the bond between the phosphate group and the molecule. Technically, that's not correct, but it's okay to have that level of understanding in a first-year life science class like this. It's not what's actually happening, and some of you might remember from chemistry that really it's the energy of the field, or the electromagnetic field surrounding the molecule, that actually holds the energy as associated with that particular molecule. But it's okay to think about it in terms of the energy being stored in the bond. So let me pause there. Is this bringing any new questions up for you before we move forward? Well, I pulled this question out of a longer AP free response question. So believe it or not, this one question is only worth 1 point. So what I'd like you to do, you can do this in the chat. You can do it privately if you don't want anyone else to see. Really, it's a 0 or a 1. Based on your answer, would you give yourself a point, or would you have been forced to give yourself 0 points? Unnecessary is not necessarily bad. Okay. Thank you to those of you who are sharing in the chat. I'm seeing a lot of zeros. So I've embedded other practice AP questions that deal with phosphate group transfer, ATP mediated energy transfer, that I would encourage you to take a look at. I've got the scoring guides in there too, so that you can always check your answer if you'd like. What I'm noticing is what the College Board is doing is they're writing one big question, and then they're creating 3 different versions of that question. Then they're making all of those questions available to teachers, which means I can make them available to you in this secure format of Zoom and Google Classroom and all that kind of good stuff. So they're in the notes, the Unit 6 notes, and I would encourage you to take a look if you need some extra practice with them. That's really how we get better, right? We don't cram, we don't memorize, we practice. So I would encourage you to do that if you think it would be helpful to you. All right. Can I clear the drawing? All right. Well, let's move forward then with the understanding that there are more practice questions in there. There's one of them right there, in fact. And begin to do some work with these functional groups. Again, with the understanding that functional groups are the parts of biological molecules that are reacting, right? As we've been talking about anabolic and catabolic processes, if we're talking about an anabolic process, well, it's going to be the functional groups, the parts of biological molecules that are going to react to form larger molecules. Remember, anabolic endergonic processes are synthetic. Things are being made from reactants. Well, of course, there's the opposite. There's catabolic processes where molecules are being broken down. But again, it's going to be these functional groups, the parts of, in this case, larger molecules that are going to be reacting with each other as those molecules are broken apart into smaller components. Okay, so I thought we could begin here with some practice. And I want to reemphasize the fact that you are not ever being asked to memorize these functional groups. Here is the chart that I believe I referenced in the Edpuzzle video. So as you work on these practice questions, I'll put you in student mode. So if you want to go back to this slide with the functional groups, you are always welcome to do that. Of course, you could take a screenshot, you could copy this slide into your own slide deck, whatever you think is best to create your own visual representation. You could print it off if you think that would be helpful. But never, ever do you have to memorize these functional groups. In fact, these next few questions are just meant to give you practice identifying some of these different functional groups. So use this as a reference if you think it would be helpful. Okay, so what I've got are some different drag your dot slides. I think it's slides 90, 91, 92, and 93. So 90, 91, 92, 93, 4 slides. Where literally you're just going to be able to drag your dot to the functional group that you're being asked to identify. And you can use that chart as a reference if you think it would be helpful. But what I'd also like to do is maybe give you 4 or 5 minutes to do that task, but also take a short break. So why don't we come back together at like 9:31? That'll give you time to practice and to stand up, take care of yourself, whatever that entails this morning. Okay, so what questions do you have before we get started with this practice? Okay. Well, let me put you in student mode. And again, the questions start on slide 90. They go up to slide 93. They're just dragging your dot slides, and you can go back to that chart if you miss or don't remember one of the functional groups. And we'll come back together at around 9:31.
Is
Sorry. I can hear you.
is the hydroxyl the OH thing, or is that different?
The hydroxyl group is an OH. Okay, so yeah, as you look at some of these different molecules, you want to be careful that you're identifying the OH, the hydroxyl functional group, and not part of a larger functional group. There is another functional group that has an OH as part of its structure, but is not a hydroxyl. Yeah, just practice. Try to get your answers in here. Get a little concerned when only half of the people are responding. Okay, that's much better. Again, you can stop at slide 93. But don't forget to take yourself a break, stand up, get a drink of water, use the restroom. All right, try to make your way back so we can take a look at these together. I hope this was useful practice. I'd love to know in the chat or out loud what you think about doing something like this. Or if you get to do these sorts of things in other classes.
We do not get breaks.
Well, the breaks, yes. It's always nice to know. I was thinking more about the dragging their dot activity.
Oh, the dragon. Oh no, we don't get that either.
You still feeling like it's lecture? Is that like a truism for most of you? Is it a universal? Is it not? All right. Why don't we take a look at these together? I hope people are there. I hope this is helpful as practice. If you're thinking remotely of considering, biology as a major in college, I'll be very honest with you. There's going to be a professor who's going to make you memorize the functional groups. It's inevitable. But I just want to reemphasize the fact that in a class like this, that is not the goal. But it is useful to be able to identify these functional groups. So having that chart in front of you or having access to that chart, I think will be helpful as we move forward in biochemistry. But let's take a look at what you came up with. I'm going to take you out of student mode here, and we'll just look at these together. So in the first molecule, you want to drag your dot to a hydroxyl group. Looks like there's some different choices here, so the answers might vary, and that's okay. But as long as you drag your blue dot to an OH, you're good. The red dot, we're trying to go for an aldehyde group. Looks like there's a little bit of confusion there, but not too much. So if I hide your responses, I can bring up my pen. The aldehyde group is one of the two carbonyl groups, and carbonyl groups all have a C double bound to an oxygen, so a carbon double bound to an oxygen. There's aldehyde functional groups, then there are ketone functional groups. This is an aldehyde functional group. Why is it an aldehyde? Well, with that C double bound to the oxygen, that carbon double bound to the oxygen, that carbon is then bound to another carbon atom, but is also bound to a hydrogen atom. So it makes it an aldehyde group. So this type of the carbonyl group, the aldehyde group, will always be found at the end of biological molecules. So much so that this is a molecule of glucose, albeit in its linear form, based upon the structure of the molecule that you see here. All right, so that looks good. Let's go on to the next one. Again, we're just checking to see how you did. This one I'm hoping was straightforward. Let's see how you did. Yeah, the carboxyl group is at the left end of this molecule. The carboxyl group is C double bond O, OH. So if we hide your responses, we can see that group over here. We want to be careful. It's not a carbonyl group because it's not attached to a hydrogen. And this carbon is not attached to a hydrogen. So it's not a ketone group because this carbon is not attached to another carbon, attached to an oxygen. So this is a carboxyl group, even though it kind of looks like a carbonyl, the C double bound O, and then the OH on the other side. In fact, that's actually why the functional group has the name that it does, carboxyl. It's like you kind of merge together carbonyl and hydroxyl. You'd have a carboxyl group. But in this case, we're just focusing on this part of the molecule, and that indeed is where the carboxyl group is located. So well done. Next one. Now we want to do another hydroxyl and now the ketone functional group. So I bet you found the hydroxyl groups. Yep, looks pretty good for the most part. And then the ketone functional group is indeed, it looks like most of you dragged the red dot to the appropriate location. It's going to be here. And why is this a ketone and not an aldehyde? Well, this carbon atom is still double bound to the oxygen, but now it's double bound to a carbon and then another carbon. So it's not bound to a hydrogen like it is in the aldehyde group. So that is indeed a ketone group, and this molecule actually would give rise to, or actually this molecule is fructose. So if you've ever heard of high fructose corn syrup, for example, this is the molecule that you are consuming. Nice job there. And then the last one. See how you did. Amino groups are in blue. Looks beautiful, nice. And then the orange to the carboxyl looks good. And then the red dot to the hydroxyl, again, we want to be careful. The only hydroxyl group that's shown here is at the bottom of the molecule. So it's down here. This is not a hydroxyl. I know it's an OH. You might assume that it's a hydroxyl, but because it's attached to this C double bond, carbon double bond to the oxygen, this entire group is carboxyl. This is not a hydroxyl. This is the hydroxyl group. Okay. So is this bringing up any questions or curiosities on your end? All right, so just a little bit of practice identifying some of the different functional groups. Why do we want to be able to do this? Well, there are lots of molecules being put together and taken apart within a living system. So there's all sorts of catabolic processes and all sorts of anabolic processes that are taking place inside of cells. And here are just a few of the reactions that are taking place inside of a typical plant cell. You know, maybe you're noticing, I tried to highlight it to the best of my ability, but here's the carbon dioxide that's entering the system. So everything that you're seeing from 1 all the way to 2, where you see this glucose 6P, those are the reactions of photosynthesis, an anabolic endergonic process. Okay, and you might notice some of the arrows that are coming off of those reactions. Well, those reactions are going to give rise to some of the other biological molecules that we're going to be exploring in biochemistry, molecules like DNA and amino acids. So I think I've given you 2 ways to earn an automatic A+ in this class, right? You can volunteer for human dissection where you are the subject. Remember, that's without anesthesia, and I will be doing that procedure on you. You can autoclave yourself. Remember, that's the sterilization equipment that we have here in the classroom. So you just got to climb inside and sterilize yourself at about, I think it's 220 degrees Fahrenheit and about 15 PSI. So you can do it that way. But I want to offer you a 3rd way to earn an instant A+. Just redraw this diagram from memory. Okay, so up to you, however you want to do it. He says sarcastically, right? All right. Well, why do we care about these functional groups? Well, again, these are the parts of molecules that are going to come together in anabolic processes like the reactions of photosynthesis, but they're also going to be the parts of biological molecules that split apart during catabolic processes. And there are 4 general categories of biological molecules where these functional groups are going to be relevant to us. There's carbohydrates, there's lipids, that's just the biological term for fats, there are proteins, and there are nucleic acids. So we're not going to even tackle these biological molecules today. In fact, we're going to be looking at these biological molecules over the course of the next week. Today, we're just going to look in general terms. How do we put these molecules together? And how do we take these molecules apart? No matter what class they might fit into, what's great about biology is you don't need to memorize like 7 different types of chemical reactions. There's just 2. One that puts molecules together and another one that takes molecules apart, no matter which type of molecule you're looking at within the living system. So let's start with the type of reaction that puts molecules together. So these are the anabolic, endergonic, synthetic processes. Okay. Well, if we're putting molecules together in biology, more often than not, what we're going to be doing is assembling monomers together. So think of a monomer as a single molecule. And in this first type of reaction, we can put those monomers together, bond them together into, well, if it only had 2 monomers, a really simple polymer. Okay. And we're going to leave it at that. We're not going to go to the level of, well, if I link 2 monomers together, is that really a polymer? Well, no, because technically you need to have at least 3 monomers that are linked together to build a polymer. So if you only link 2 together, you only really have a dimer, but who cares? Who cares? We're linking 2 molecules together. It's bigger than what we started with. We can call that a really short polymer, okay? So what's great is any of those biological molecules, carbohydrates, fats, proteins, nucleic acids, they're all gonna be assembled in the same way. They're all going to be assembled from their monomeric constituents, so from single molecules, to build larger polymers. All right? And the reaction that takes place in order for that to be accomplished, it actually has two names. So I like the term condensation reaction, but I'll be very transparent. The College Board tends to use the synonym dehydration. So don't let that confuse you. It's just two different terms for the same type of reaction. I like condensation because in my mind, I'm taking two molecules and I'm condensing them into one. You're going to see very quickly why dehydration also works as a descriptor for this type of reaction. Right? So let me pause there. Does it bring up any questions for you? Anything at all? Okay. So in this reaction, this dehydration reaction, in this condensation reaction, in the example that I put up on the slide, we're actually starting with a short polymer. So we've already linked 3 monomers together, the purple circles. And what we're going to do in this reaction is add 1 more monomer to that short polymer chain. So we'll have a polymer that's 4 monomers long as a result. So what's happening during that reaction? Well, looky here. This is why the functional groups matter. So here's this monomer coming in. We have no idea what it is. It doesn't matter. It's a generic monomer, but it has a hydroxyl group. Okay. The short polymer has a hydrogen atom associated with its structure. So again, we don't know exactly what that hydrogen atom is a part of, but maybe it's part of the aldehyde. Maybe it's part of a carboxyl group, right? Maybe it's part of another hydroxyl group, and for whatever reason, the author, the illustrator, didn't add the oxygen in there. Okay. But we do see that there's an OH on one molecule and a hydrogen on the other. Well, the reason why these reactions are known as dehydration reactions is because when this monomer is added to the short polymer chain and the new bond forms between the short polymer chain and that monomer, 1 molecule of water is produced as well. That's why these are called dehydration reactions, because a molecule of water is coming out, right? We're dehydrating the polymer as it's being made. Now, keep in mind, in reality, all of these reactions are taking place in water, in aqueous environments. So, we're not drying out the system or drying out the molecule in a meaningful way. We just call these reactions dehydration reactions because water is a product of the process. Okay. But what I want you to notice is that it's these functional groups that are actually reacting with each other to form the molecule of water, but also to form the new covalent bond that links the molecules together. Okay. Shall we try some practice questions? Thumbs up, thumbs down. All right, so this is an AP multiple choice question. So again, a process you've never seen before. Which of those answer choices makes the most sense to you? About halfway there. All right, looks like most people have had a chance to respond. Let's take a look at how you did. So it looks like the leading vote getter is C. That is not correct. Okay. It's not correct first because we didn't talk about a hydrolysis reaction yet. So now your answers are all going to flip, right? And we're not breaking bonds in that reaction. We're actually forming a bond. So while you don't know the name of that bond yet, you will by the end of the week. You are forming a peptide bond, but really what you're forming, if we go back a second, is a new covalent bond. So we can actually illustrate how that bond is forming between these two monomers. So this is monomer number 1 up top. And you can think of this as monomer number 2. Okay. And we're going to link those two monomers together into a really simple polymer, a 2 monomer long polymer, if you will. Okay. So again, to review, we've got 2 functional groups. There's the carboxyl, and there's the amino group. We're going to link these 2 monomers together, so we're going to use that dehydration reaction, that condensation reaction. Okay. Well, where's the water coming from? Well, in a reaction like this, it's coming from the OH of a carboxyl group and the hydrogen of the amino group. So OH and H, that's where the water is coming from in this reaction. And then as those 2 monomers come together, a new chemical bond, a covalent bond, is forming. That bond is right here. It is a peptide bond, but you don't need to know that in order to answer the question, right? This is a dehydration reaction where a new covalent bond is forming between these 2 molecules. And yes, D is by far the correct answer here in a case like this. So I want to check in with you before we move forward. I've got one more practice question before you sign off. A lot of people chose C and not D. Are there questions, concerns about why it is D and not C? Does my explanation help? Still monitoring the chat. Okay, I'm gonna trust you. If you have questions, you're gonna ask. Let's try another practice question. Not this one. All right, this one's a little bit different, not multiple choice. You're gonna have to enter a number as your answer. But let's say you're gonna make a polymer, a polysaccharide. It's a type of carbohydrate. So you're making a polymer from 25 monosaccharides. The monosaccharide is the monomer. So 25 of them. You're going to put those 25 monomers together to make the 25 monomer long polymer. How many molecules of water would be synthesized as product in order to make that polymer? If you put 2 monomers together, you get out 1 molecule of water. If you added another monomer to that 2 monomer long chain, you get another molecule of water out. How many molecules of water would you get out if you linked 25 monomers together? It's okay if you're not sure. We've got time to go over it. Try to put something up there. See how you did. So, looks like it fell into two categories. What we're looking for is 24. And a lot of people answered 24 correctly. Why is it 24? Well, quickly, again, if I had two monomers and I link them together via a new covalent bond, I'm going to get one molecule of water out as a product. If I took that molecule and I added another monomer to it, it gets a little busy, I would get another molecule of water out. And I can keep doing this. I could hypothetically draw it for all 25 monomers. That would be really taxing. But really, for every bond that forms, I'm going to get out one molecule of water. So if I want to link together 25 monomers, I need to form 24 bonds, covalent bonds, to link those monomers together. And if every bond that forms, I'm going to generate one molecule of water as a product. So in this case, indeed, it would be 24 molecules of water that are going to be produced to link all of these monosaccharides, these monomers together to make the 25 long polymer. Okay. All right. Well, now we're just about out of time. So we'll save the other type of reaction for next time. And I've got lots of practice questions that we can take a look at. So, but before you go, if you could, in this last minute, what's for you the most important takeaway from this lesson? Thinking back to what we did today. We did some work with the functional groups. We did some work with the type of reaction that links molecules together based on those functional groups. Add your thought, your reflection, and as soon as you have, you're welcome to sign off. As always, thank you for being here. I'll see some of you face-to-face on Wednesday, I think. Others I'll see you on Zoom, of course. Please be well. Take care of yourselves. Bye, everybody.